Hello friends, today we will discuss about immunity. Okay, so immunity, as you know, it is a defense mechanism of our body. Basically, immunity is classified into two types. What are that two types? Innate immunity and adaptive immunity. Innate, that is by birth, okay, and another one is adaptive immunity. It is also called as acquired immunity, right? Today we'll discuss about this adaptive or the acquired immunity. Again, this adaptive immunity is of two types. One, it is known as cell mediated immunity. Cell mediated immunity. Another one, it is known as humoral immunity. Okay, so in this today's lecture, we'll first concentrate on this cell mediated immunity. In the next video, we'll discuss about the humoral immunity. Before going in detail about this type of or the mechanism of the cell mediated immunity, I would like to give some background regarding the lymphocytes. So lymphocytes, very important cell of WBC. So you know, WBCs are basically classified as granulocytes and agranulocytes, and granulocytes are neutrophil, eosinophil and basophil, and agranulocytes are lymphocytes and monocytes. So basically it is a WBC or a leukocyte. So this lymphocyte will help in development of either cell mediated or humoral immunity. Again, this lymphocyte is of two types. One is known as T cells or the T lymphocytes, another one is known as B cells or the B lymphocytes. So these B lymphocytes will help in the development of the humoral immunity, and T lymphocytes helps in the development of cell mediated immunity. So today we'll discuss that cell mediated immunity, how these T lymphocytes help in the development of cell mediated immunity. Okay, so to describe this, we'll take an example of any antigen. You know, antigen is any foreign substance, either bacteria or a toxin or any other chemical compound which enters in our body, which stimulates our immune response. Immune response or immunity or adaptive immunity is mainly due to lymphocytes. So they will stimulate the lymphocytes, either T lymphocytes or B lymphocytes. So when B lymphocytes are stimulated, then it leads to the development of antibody formation that leads to the killing of that bacteria by the antibodies, and that mechanism known as humoral immunity. Or T lymphocytes are activated, then T lymphocytes kill that bacteria. That mechanism known as cell mediated immunity. Hope it is very much clear what is cell mediated immunity, what is humoral. Cell mediated immunity is through T cell and humoral immunity is through B cell. Okay. Now, okay, what is T cell, what is B cell? Okay, what is the difference between this T and B lymphocyte? So let me take some time to explain this before going in detail about the mechanism of cell mediated immunity. So see, in the bone marrow, all these cells are produced, that is the hemopoiesis. Hemopoiesis is RBCs, WBCs and other like platelets, and developed basically in the bone marrow, right? Let's consider this as a bone marrow. So in the bone marrow, the hemopoietic cells, firstly the immature cell or the blast cells will be developing. Suppose this is a developing lymphocyte. So basically this development starts before birth, that is in the intrauterine life, that is in the fetal life itself, right? So in the bone marrow, suppose the lymphocytes are developing. So when they develop, they enter into the blood stream or the blood vessels, and through the blood vessels it will go to the different organs. Those lymphocytes, during the time course of development, that is, this lymphocyte is formed, but still it requires certain pre-processing or some training. That lymphocytes which enter thymus. Thymus is an organ, okay, so which enters the thymus, and they get further pre-processing or development or training in the thymus tissue. And once they get training or development in the thymus tissue, they come out, and again they come out into the circulation and circulate in the blood vessels, and or get located in the lymphoid tissues like lymph nodes, spleen and thymus itself, or adenoids, tonsils, or mucosa associated lymphoid tissue in GI tract. So they get stored there and in the circulation itself. That lymphocyte is known as T lymphocytes. So what is T lymphocytes? The lymphocytes which are educated and trained in the thymus is known as simply T lymphocytes. Now what about B lymphocytes? See, B lymphocyte was first discovered in the birds, in the organ or it is in the bursts of birth, is known as bursts of Fabricius. So in the bursts of Fabricius, this B lymphocytes was first identified. That's why the name given was B lymphocytes. Okay. Now these developing T lymphocytes which enter into the blood, again it will go back here in the bone marrow, and bone marrow, in the bone marrow itself, where there is a development takes place in humans, it will develop in the bone marrow, and again that will enter into the circulation and goes to the lymph nodes and store there or in the other lymphoid tissue. That lymphocytes are known as B lymphocytes. So what are B lymphocytes? Their training and development takes place in the, or preprocessing takes place in the in the bone marrow, and T in the thymus. Now we have millions of lymphocytes all over in our body, and mainly they are situated in the lymphoid tissue like lymph nodes, spleen, thymus. Already I have enumerated all the things, including the circulating blood. Now each T lymphocyte is different from its other T lymphocyte. Each B lymphocyte is different from other B lymphocyte. Let me highlight this point very because it is very important. So see, there will be many T lymphocytes, but each T lymphocyte will be different from each other. How? Or because of the configuration of their surface receptor. On the surface of the T lymphocyte, there is a protein molecule. This is known as simply T cell receptor. Now this T cell receptor on the different T cells will be different in configuration. Okay. For example, this T cell receptor can bind to this rectangular type of antigen. Okay, the another T cell receptor can bind to the square type of antigen. So this T cell receptor, the configuration will be like this. Okay, and this T cell receptor can bind to the circular shape. So the configuration will be circular shape. The T cell receptor will be like this shape. So each T cells will have the receptor which will be having a different configuration or simply different shape. So when a particular antigen of that configuration comes, then only it can bind. See, this triangular shaped antigen can't bind to the square shaped or circular shaped. So there are millions of such T lymphocytes which are designed in such a way in our body, the beauty of God, the beauty of nature, in such a way we have been designed that in the lymph nodes, consider just a lymph node, in that T lymphocyte with the B lymphocyte, there will be several T lymphocytes, and on surface of T lymphocytes, these T cell receptors are there and they are different. For example, one T lymphocyte can bind only to the typhoid bacteria, that is the Salmonella typhi. One T lymphocyte can bind only to tuberculous bacteria, that is this one, tuberculous bacilli. Okay, similarly other for dengue virus, whatever, but this can't bind to this. Okay, there are so many T lymphocytes, but they are different. T cell receptor is different for, and they are designed to bind to specific antigen only. Similarly, B lymphocytes also. So on the surface of the B lymphocyte also there is B cell receptor. Okay, like T cell receptor, that there is a B cell receptor. Okay, but this B cell receptor is basically an immunoglobulin, either IgD or IgM. So again, the configuration of this varies. So we'll discuss about these B lymphocytes when we discuss about human immunity. Right now, just concentrate on the T lymphocytes. I have taken much of your time to explain this basics because it is very much essential to understand the today's topic of cell-mediated immunity. Now come to the today's topic about cell-mediated immunity. It is a 5 marks very important question for your theory examination in the subject physiology, also cell-mediated immunity. So also in the subject pathology or microbiology, this question they have asked several times in the theory exams, also clinically also it is very much important. Now with this background we will start this cell-mediated immunity. What exactly it is hope you know now by the activation of T lymphocytes, how T lymphocytes are activated and how it will kill the foreign organisms or bacteria. Now T lymphocytes are of T type, for 4 types, T lymphocytes are of 4 types. What are that? One, it is called as T helper cells. T helper cells, it is also known as CD4 molecules. Second type is cytotoxic T cells. Cytotoxic T cells, it is also known as CD8 cells, or also it is known as killer cells or killer cells. We'll come to the individual this T lymphocytes. Okay, but right now just try to know what are the different types of T lymphocytes. T helper cells, cytotoxic T cells, then T suppressor cell or regulatory cells, suppressor cells, or simply T regulatory cells. Regulatory cells. These are the different types of T cells. One more is memory T cells. Okay, there are basically 4 types of T cells you are supposed to know, and when you write in the examination, you should write about this, explain about these 4 types of T lymphocytes. Now, we'll try to explain this first. Basically, what is cell-mediated immunity? When any antigen is any foreign substance like microorganisms or chemicals or toxins which enter in our body. In response to that microorganism or toxin, our body, especially the lymphocytes, which cell will activate? Lymphocytes. In this connection of cell-mediated immunity, T lymphocytes will get activated and they will kill the bacteria. So now today we learn we just imagine a bacteria has entered in our upper respiratory tract, either through the nasal route or the oral route. Through air, it entered in our body and it lodged in our pharynx and it causes pharyngitis, that is inflammation of the pharynx. Now, how that bacteria is killed by T lymphocytes, that's what we are going to study now. So just consider this is a pharynx, okay, and this bacteria, many bacteria, this red color bacteria, these bacteria entered our pharynx and they are causing inflammation. Now first line, what will happen is, at the site of inflammation means tissue response to an injury. Basically, the details we'll study in some other video about inflammation and all. So there is the redness and all. So first thing is the most important cell, that is the WBC, which will come out from the this one blood to the tissues, to the pharynx, is neutrophils. So what these neutrophils will do is they will engulf this bacteria and they will phagocytize. Phagocytize means cell eating. They will eat this bacteria. Okay. So this neutrophil at this site, they will enter and they will eat this bacteria. With the help of the lysozymes and the other chemicals, it will kill this bacteria. Okay, phagolysosome, they will kill it and this bacteria is destroyed and their products, that is the bacterial antigens, are released into the blood circulation. Bacterial antigens, okay, are released into the circulation. Now this bacterial antigen, through the lymphatics or even through the blood vessels, it will go to the nearest lymphoid organs, for example tonsils, adenoids, or even lymph node. Now consider this as a lymph node. So this is pharynx, where there is, so ultimately what we are learning is, through this discussion, in the pharynx bacteria has entered and this bacteria need to be killed by T lymphocytes in the cell-mediated immunity. How it will kill? Let us discuss. Now, this neutrophil which is releasing this bacteria, it will go either via lymphatics or through the blood to the nearest lymph nodes. Now, this lymph node, in this lymph node, already there are lymphocytes are present, both B and T. Few will be T lymphocytes, few will be B lymphocytes. Now, this antigen, bacterial antigen, which comes into the lymph node, now they will try to bind to the T or B lymphocyte, but T lymphocyte can't bind directly the free bacterial antigen. B lymphocyte is a humble cell, it can bind directly to this one. B cell receptor can bind this free antigen and it will lead to the production of antibodies, that is human immunity. But for the T lymphocyte activation, for cell-mediated immunity, it requires one more cell. The other cell which is important here is macrophage or B cell or dendritic cell. These cells are known as antigen presenting cell. Antigen presenting cells. So what are the antigen presenting cells? Examples are macrophage, B cell, or dendritic cell. So these cells will present the antigen to the T lymphocyte. They will go and take the antigen to the T cell, then only these T cells in the lymphocytes will get activated, or else it will not. B cells directly. Now We'll take one example of macrophage. Now, how this macrophage are present in the blood, also in the tissues also. This macrophage can engulf and it will go via lymphatics to the lymph nodes, and they can present the antigen to the T cells, or in the lymph node itself there will be many macrophages present. Okay, they will take this foreign bacterial antigen either from here or here, and ultimately they will present to T cells. Now see that mechanism, okay, that is most important, how exactly this macrophage will present the T cell, and T cell get activated, and this T cell will kill this bacteria which is there in the pharynx. How it will kill, let us see, or it is circulating in the blood or in any other site. So consider this as a macrophage. Okay, what is macrophage? Macrophage is nothing but a monocyte. Right, monocyte is a WBC or a leukocyte. Monocytes, which comes out of the circulation and settled permanently in some tissue, that is known as macrophages. Simply, okay, we'll discuss about that tissue macrophage system some other video. Right now, it is basically derived from monocyte, which is a WBC. Now, this macrophage will engulf the bacteria again by the phagocytosis. This is the bacteria. Right, and again it will, like neutrophil, it will try to spill the bacterial antigen outside, but it will not spill directly. It will hold the bacteria on the cell membrane. How it will hold, let us see. On this nucleus, you know, there are DNAs, genetic materials will be present, chromosomes will be present in the nucleus. So on the short arm of the chromosome number 6, consider this as chromosome number 6. On the short arm of the chromosome number 6, there is a gene present. This gene, which is present in the nucleus on the short arm of the chromosome 6 of the macrophage or any antigen presenting cell, it will code for the production of one protein by the process of transcription and translation. Hope you know this central dogma of biology, that is this DNA, they will form mRNA, is known as transcription, and again this translation, when proteins are formed, and that protein which is formed, it will be coming and it will be present at this surface, and that protein which is coded by this chromosome 6 of the antigen presenting cell, it is known as MHC, and more specifically class 2, major histocompatibility complex class 2 molecule. Again, what is this MHC and all, we'll discuss in some other video. Timing, friends, just concentrate here because the lecture will be very lengthy if I go and tell details about this, what is class 1, class 2 and how it will helpful, and right, me just remember, on the antigen presenting cell, major histocompatibility complex protein 2 molecule is coded by the chromosome number 6, and it will present on the surface. Now this will hold the bacterial antigen. What neutrophil was doing? Neutrophil was just spilling that antigen and it was going to the nearest lymph node or lymphoid organ where B lymphocytes are taken, but T lymphocytes can't take directly. T lymphocytes want an antigen to be presented by the antigen presenting cell. What are the three types of antigen presenting cell? Macrophage, B cell and dendritic cell. Dendritic cells are more common. So this macrophage or the dendritic cell will present or hold the antigen with the help of major histocompatibility 2 molecule. Now this macrophage with the help of major histocompatibility 2 molecule will hold the antigen and take to the nearest T lymphocytes in the lymph nodes. So in the lymph nodes there are so many T lymphocytes. They will go and present this to T lymphocyte. But on the surface of the T lymphocyte, as I said you already, we have discussed that on the surface of the T lymphocyte there are T cells receptor. What are there? T cell receptor will be there. Now different T cell receptors will be there. As I said you, there will be many T cell receptor D lymphocytes. Those who are destined to combine with this type of antigen, they will only get selected. Rest all T lymphocytes are not activated. Out of this, so many thousands or hundreds of T lymphocytes where this macrophage will go and present to the T lymphocytes, one T lymphocyte or 2 or 3 will get selected and they will only can bind to that particular antigen. No other lymphocytes can bind. Okay, each T lymphocyte is designed to bind a specific antigen. Okay, for example, if it is a typhoid, Salmonella typhi bacteria, it can bind to only one type of T lymphocyte. Okay, another tuberculosis or other bacteria can bind to the other. T lymphocyte. So one type of the T lymphocyte is now selected, which has the same configuration, that is T cell receptor, which can fit to this bacteria, and most importantly presented by antigen presenting cell with MHC2 molecule. They get selected, and this process of selection by the T lymphocytes, it is known as T cell clonal selection. So one clone of the lymphocytes is selected. Okay, not all T lymphocytes which are present in the lymph node, only one clone. Now, as I said, you T lymphocyte is a very delicate cell. It will only accept the antigen and is present by MHC2 molecule. Now, how T cell will recognize that this antigen is attached to the MHC molecule? On the surface of the T cell, there is one more protein or receptor. This protein is named as CD4. Full form of CD is cluster of differentiation 4. Okay. Now, this CD4 protein will recognize this MHC2 molecule, whether this antigen is coming with the help of MHC2 or not. Okay, this will check it. So once it confirms that yes, there is a MHC2 molecule brought by the macrophage or the antigen presenting cell, consider this as a macrophage and this as a T lymphocyte. So when CD4 molecule binds with the major histocompatibility 2 molecule of the macrophage, so this will now, when it is this T cell recognizes the major histocompatibility 2 molecule, then only this antigen molecule of bacteria will bind to the T cell receptor. This is T cell receptor, this is CD4 molecule. They will bind. After binding of the antigen to T cell receptor, the inside there is one protein known as CD3. This protein gets activated and it sends signal to the nucleus of the T lymphocyte. Now that message has been gone to the nucleus of the T lymphocyte that the antigen has come. Now you need to activate and destroy the bacteria wherever it is present. All these things are happening in the right now in the nearest lymph node, okay, where T cells are there. So T cells now get the message through macrophage directly. Antigen bacteria, T cell won't accept the message, same like unlike B cells. Now after getting this, T cell will get activated. Now this macrophage will send the signal by other routes also. So it will send the signal, the nucleus will produce certain chemicals. One such chemical is interleukin 1. You know, this is a leukocyte. Monocyte is a leukocyte. Monocyte which has become the macrophage. Okay, and this is also leukocyte. So when there is a communication between two leukocytes, it is called as interleukins. Okay, so interleukins 1 will go and communicate with this T lymphocyte, and again it will send the message to the nucleus of the T lymphocyte now that antigen is there, the foreign bacteria is there in our body. Now you get activated and produce something. Not only this, further macrophage will again give some information with the help of one surface protein. Okay, what is that surface protein? So this red color what I am drawing, this surface protein is known as B7. Again it will bind to one more membrane protein which is present on the T cell. It is known as CD28. So by 3 waves it will give information to the T lymphocyte. First by direct binding to T cell receptor, second by interleukin 1, third by this B7 protein combining with the CD28 molecule of T lymphocyte. Now all this information goes to the nucleus of the T lymphocyte. Now this T lymphocyte, what it will do is it will produce a very important interleukin 2. Now this interleukin 2 further go and activate interleukin 2 receptor and further activates the nucleus of the T cell, and these T cells will undergo now mitosis, and many such T cells are produced. And this process of mitosis or proliferation or multiplication of the T cell, it is known as T cell clonal proliferation. Clonal proliferation. Earlier it was clonal selection, that same group will again multiply. That is known as clonal proliferation, and again it is due to production of interleukin 2. Now, this type of T cells, on the surface of which there is a CD4 molecule, this type of T cell, it is simply known as, what is this known as? T helper cells, because it will help other cells. How it will help, I'll let you know. First, remember what is T helper cells? T lymphocyte, simply on the surface of which there is CD4 protein, it is known as T lymphocyte, T helper cells. Okay. Now this T helper cells, as you know, they produce interleukin 2. Now this interleukin 2, which is also known as lymphokine. So lymphokine, what it will do is, it will go and stimulate the other T cells. Other variety of T cell, that other variety of the T cell, it is known as cytotoxic T cell. How this cytotoxic T cell is different from T helper cell is, on the surface of the cytotoxic T cells, the membrane protein is CD8. Instead of CD4, it is CD8. So those T lymphocytes on the surface of which there is CD8. CD means cluster of differentiation, full form, cluster of differentiation type 8. CD8, that type, it is known as cytotoxic T cells. It is also known as killer cells. Why it is called as killer cells is because ultimately these cells, these T lymphocytes, which are activated by the production of this interleukin 2, this interleukin 2 which is produced by the T helper cell. That's why the name given T helper cell. T helper cell, it will help the other lymphocyte like cytotoxic T cells to multiply and kill the bacteria. So ultimately cytotoxic T cells will stimulated by interleukin 2, and there is multiplication of these cytotoxic T cells, and more number of the cytotoxic T cells, that is CD8 cells or the killer cells, are produced. Okay, now these CD8 cells, which are more number, they are produced in this lymph node itself. They will go into the blood circulation, and they will go at the site of pharynx or where the bacteria is there, or even in the blood circulation wherever the bacteria is there, and they go and kill it. How it will go and kill it, that is the mechanism. Let us see. Now consider this as a bacteria, this as a bacteria, and the CD8 cells or the cytotoxic T cells, they will go and bind to the bacterial cell wall or the cell membrane, or even the cells which are infected by the bacteria. Okay, now what they will do is, they will bind it and they will release one chemical. That chemical which is released, it is known as perforins. Perforins are nothing but the hole forming proteins. So ultimately they will release certain chemicals, perforins. That perforins, what it will do, they will produce as holes on this bacterial cell wall or the cell membrane. Okay, now these holes will causes the fluid or the water or the sodium which is present in the tissue fluids to come inside the bacteria, and the bacteria or the cell which is infected bacteria will get swelled up, and it will ultimately burst and it will die. Okay, this is one of the mechanism of killing of the bacteria by this cytotoxic T cells, by releasing perforins. What are perforins? Hole forming proteins. Then secondly, not only they will form the perforin, they will also form some other chemical. These are known as cytotoxins or cytotoxic substances. They are, that is also released by the CD8 cell, and they enter into these holes which are formed by the perforins, and they will kill the bacteria simply, okay, by cytotoxins or cytotoxic substances. Then the third mechanism is also, it will activate and kill the bacteria by the process of apoptosis. Okay, we have discussed about this topic apoptosis in detail. Please go back to our playlist in the general physiology or just physiology, where we have discussed in detail about the mechanism of apoptosis, that is the programmed cell death, how this cell death occurs. Okay, now just to remember the mnemonic cap: C for cytotoxins, A for apoptosis, P for perforins. So through this cap mechanisms of cytotoxins, apoptosis and perforins, cytotoxic T cells kills the bacteria. Now this is simply killing of the bacteria by the activated T cells, and it is known as cell mediated immunity. It's not over, but still, what is cell-mediated immunity? Killing of the bacteria by the activated T cells. We have said you the story how this bacteria is getting T cells are activated by the antigen presenting cell by three ways, either directly, either by B cell proteins or interleukin 1, which will ultimately produces interleukin 2, that is the T helper cell. It will help the multiplication of what cytotoxic T cells by the interleukin 2. Not only it will produce interleukin 2, it will also produce interleukin 4, interleukin 5, interleukin 6. Now these interleukins or the lymphokines will help the neighboring B lymphocytes. So it will help simultaneously. T lymphocytes are activated, even the B lymphocytes. We'll discuss about that B lymphocyte story in humoral immunity. Now these B lymphocytes which are present in this lymph node, they are exposed to the antigen and they are also multiplying. So for the growth and multiplication of this B lymphocyte, this interleukin 4, 5 and 6 are essential, and that's why these lymphokines are known as B cell growth factor or B cell differentiation factor. Okay, they help in development of B lymphocyte, and B lymphocyte develop into plasma cells, and from plasma cells antibodies are formed, and these antibodies will kill the bacteria, that is humoral immunity. We'll discuss later. Right now concentrate here. So interleukin 4, 5 and 6 are formed. Now this interleukin 2, as I said you, it will activate the cytotoxic T cells and help in multiplication of that, and that ultimately kill that. There is a third form of the T lymphocyte which is known as T suppressor cell or T regulatory cells. In addition to the CD8 cells, these cells are also one different variety of these T lymphocytes, that is T suppressor cell or regulatory cells, are activated. What they will do is, sometimes these cytotoxic T cells, after killing the bacteria, they may damage our own tissues. They may kill the surrounding neighboring healthy tissues also. To check the production of this, they will inhibit T suppressors. They will suppress the production of CD8 cells or even itself T helper cells. Okay, so they will suppress or regulate the production of this killing. That's why it is known as T suppressor cells. Okay, now finally, few of the T helper cells are formed as T memory cells. What are these T memory cells? You know, these T helper cells, they will multiply. Okay, they will multiply by clonal proliferation stage into many. So all these T helper cells will not develop they will help the other T lymphocytes or B lymphocytes or macrophages to kill the microorganism, but few cells will remain dormant, inactive right now, and it will be stored in our body through the blood circulation. They will go and store in certain tissues or lymphoid tissues or in the blood circulation also. When the same antigen enters in the next time, already some these activated cells which are in the dormant stage, they will get activated fast and they will kill the bacteria for the second time. So this type of T cells are known as T memory cells. Okay, they will be in the memory of the immune system. Similarly, we have B memory cells. So we have totally 4 types of T cells. T helper cells, which is very important, CD4 cells. In AIDS, acquired immunodeficiency syndrome, these CD4 cells are less in quantity. That's why they are not able to release this interleukin 2, and ultimately the CD8 cells are unable to kill the viruses, right? That is, the cell-mediated immunity is less, and also this T helper cells help the B cells by interleukin 4, 5 and 6, but that is also not there because of the less quantity of T helper cells in the AIDS. Okay, just to summarize what we have studied today, we have studied about this adaptive immunity, cell-mediated immunity by these 4 important cells. Hope you like our video. If so, please subscribe our channel Doctor's Corner and stay tuned with us for similar type of lectures on all the subjects of MBBS, especially now physiology and other subjects also. We will try to make it frequently. Thank you.